and I welcome you all once again for this wonderful lecture. I'm sure, and let me hand over a small memento on this occasion for to her for gracing the occasion and agreeing to give a talk. Thank you.
Thank you. Thank you. Thank you all. Thank you to ICTS and the Planetarium for hosting me and for giving me this invitation to present today. I'm really delighted to be here at the Planetarium. It's such a beautiful campus, and I think my son also did part of the REAP training over here, so I'm very grateful to all the people who are part of the Planetarium who made this possible as well. So, so today I thought I'm going to talk, like the director said, I have wide range interests which go from nonlinear elasticity all the way to how insects cut and bore through substrates. And since this is a public talk, I won't go into very specific details, but what I thought I'll do is to try and show you how fascinating the world of natural biomaterials is. And just to preempt this, somebody, when I said I'm giving a talk on nature's rubber proteins, they said, I hope you know that the rubber we get from trees is not the real protein. So I want to start by telling you that we draw some inspiration from that part, but I'm going to talk to you about 3 specific systems. And to motivate this, I'll begin with one video. All the videos are from YouTube, and I would have given the citation there. But what you're going to see is, the first one is the amazing jump of a flea. So a flea is a small insect that sits on cats, on dogs, and so they have this ability, and you'll hear it, so that
100 times faster than the blink of an eye. But it takes more than muscle to move that fast. The flea can compress itself like a spring, thanks to a rubbery protein called resilin in its legs and body. First, the flea pulls in its oversized rear legs and locks them in place. It squeezes and flattens its body, squishing the resilin and building up energy for the jump. Then, it releases the energy all at once. The flea tumbles through the air, legs extended.
And just in case you didn't catch how cool that was, the flea is making a jump which is about 150 times its body length. So just to give you a perspective, if I were able to jump, I would scale the height of the Eiffel Tower. And of course, I can't do that, but a flea can do that. And so the question is, how is the flea able to accomplish such a fantastic feat? And it's, no pun intended, the way this works is, at the base of its leg, there is a small pad which is made of a rubbery protein, and it seems to push down, and that uses it as an energy storage device. And as soon as it lets go, all the energy which seems to be trapped in that small unit seems to be given back to this flea, which is able to do this thing. So early on, people, and I'll talk a lot more about this protein in a little bit, but this is the potential model of how this works. So there's the tibia and the femur, or the leg parts of the insect, and it crouches and pushes down on it, and as soon as it's kept this structure under tension, and under compression, and it lets go, and then the flea is able to make this jump. Just to tell you that this is not the only interesting rubber-like protein which one sees in the insect world. So here what you'll see is a mollusk. It's a bivalve. So any of us who have seen, who have gone on the beach and see these lovely shells, they come from bivalves. So there are 2 parts to it. And there's a small mollusk which sits inside. And this is what you're going to see in this video, is how does this animal which lives in the ocean actually locomote, and how does it escape predation. So, so what you'll see is, So it's moving really fast, actually, if you think about it. Now try and think of this water as being extremely cold, something like an Antarctic water. And this is the kind of quick motion that the animal needs to make. And again, it seems to move really fast. And the question is, how does it do it? And just to tell you a little bit, at the base of those valves is another small pad of rubber-like protein which is present. So the third example is closer to home. This is the heart. So the heart, as you see, is a pump. So the mechanical engineers love pumps. So there was no way I could not put one such picture. So what you have here is blood which goes from the left ventricle. It gets pushed through this unit, if you can see my cursor. So it goes all the way here up through this structure called an aorta. And then it goes to the rest of the body. But while the pump is working about 72 beats per minute, what we want to ensure is that there is continuous blood in the rest of the circulation. And the job of the pump is not just to push the blood, but for that energy to be given to the blood so that it can travel further downstream. So these are the challenges that we face as vertebrates. And the way we are able to do this with just an example, if we are to run this thing without, so if this is your heart pumping, and that's the liquid which is blood flowing out, and it's going to be very choppy, where the blood starts and stops, and like it says, it's squirting. But as soon as we have this bulb, which over here is now that aorta structure that you have here, and you'll notice that the blood is continuing to flow very uniformly. And it's able to do this because the actual job of taking the blood is getting, so the aorta is getting stretched out. And to give you a sense of how much it stretches out, to almost 80% of its normal size. So that's the amount of deformations that these structures have. And it's able to do that because of another protein, which is called elastin. And it acts as a it acts as an energy storage system. And it's able to not just stretch, but as it returns back to its normal size, all that energy is given to the blood. The other job of the aorta is to dampen this pulsatility that you have built inside the heart. So because otherwise it was not very efficient as a pump. So this system is called a wind kessel. And I'll come back to this a little later to tell you. So these are three broad examples in three different systems. First, we started with insects, and I gave you one example of how fleas jump. Now, that's not the only place where it's relevant, but it is also present in at the wing hinge of dragonflies. So if you've seen dragonflies which hover or dragonflies which migrate, they're able to do these remarkable tasks several times because they are able to use these materials as energy storage devices. So the three classes are resilin, which is a compression energy storage pad, elastin, which is very relevant, and you'll see a lot of studies for people who like to look at elastin because it has a disease component associated with it. But it's present in our skin, in our arteries. In women, it's present in the uterus. So as you have a baby, there's a lot of elastin which is made, which allows the structure to become large. Abductin is singularly found in mollusks like the scallops. All of them have these characteristics that they will exhibit long-range elastic deformation. So you can push it and it undergoes more than 80% or 100% deformations. And the more interesting thing is it exists as a one-phase amorphous, because anyone who likes to study proteins first tries to understand what is the structure of this protein. So here are three classes of proteins which are amorphous. That means you can't actually have a structure to these proteins. So the question which people have been trying to understand is, what is the mechanism of elasticity? How are these really small materials able to do these fantastic tasks? So if we try and study biological materials, so we like to pull them. And most people ask us, so why do you really want to pull them? What do you gain from understanding this? And the reason is because we get to understand a lot about how the structure behaves under many of these conditions that we put. What you see on the left-hand side is a piece of elastomer. Elastomer is a rubber-like material which is cooked up in the lab. And as we pull it, you'll notice that the central gauge region, where in the dumbbell region, the blue color part has thinned out and it has become very long. And we can look for length changes between these two. And when we plot this graph, so the way it works is we have clamped it in a system and we pull it. So we are putting in some kind of forces. And we take that force and we divide it by that cross-sectional area, and we call it a unit called a stress. A stress is nothing else but force per unit area. Of course, one can ask the question, which force, which area? So very soon the picture starts becoming very murky because you'll have 3 different forces and many different areas. So these are actually what are called tensorial quantities. For now, we will just assume that it's a force per unit area to simplify our relation. Strain is change in length upon original length. And the reason why we go from force displacement to stress strain is to take care of the geometric parameters. So for example, in my lab, I could make a really small material and you could make something really large. But to non-dimensionalize it, we try and work with stresses and strains. And when we plot this graph, stress versus strain, what you see is the initial part of this curve is very linear. And that's very good because we can take a slope of that and we call it a Young's modulus. A Young's modulus tells us something about how the material behaves. So I will use this term quite a bit through my talk. It's called a modulus. This is a cartoon of a typical engineering material. So with that, you'll see that there is something called an elastic limit, which you'll see for steels. Most engineers love steels, so we start right there. And it gets to the maximum stress, and then finally it will fracture. But the energy under this curve is what we will broadly call as toughness. There are many other measures, but it gives you a sense of the energy which is involved in doing this process. But things are not so simple for biological tissues because this very nice straight line graph that you saw for steels doesn't really work for biological materials. And this is the graph that we have got for that material, which is an elastomer. And so you'll see that the graph is highly nonlinear. So the question is, where do we calculate that slope? And at each point of this x-axis, you can get a different slope. So our relationship between stress strain is a little more tricky. 2 is there are more challenges because it's not as simple to clamp samples to systems like this. And how do you measure this length changes? For steels, we can actually clamp some transducers and get reports. But here we have to make all these dots, you have to have a camera, and others, those are other artifacts of the system. So things are a little challenging, but we are able to get equivalent measures. So what happens when we start plotting these, what I will call stress, strain data for various materials? What you see in yellow is stainless steel, and you'll see that the y-axis is broken at many points because I couldn't fit every kind of material on the same graph. So I put my favorites over here. So you have steel right up there. So it goes through that straight line, and you'll see very little deformation in the sample on the x-axis. And then it goes to this part, and it goes to ultimate tensile strength. Cork is another nice material. A lot of us use cork. And so in the wine bottles and stuff. So you'll see it also has a behavior which looks a little bit similar to that of steels. And then there is bone. So bone, you'll notice, gets this very straight line, but then it's a flat response, and then it fractures. If we start adding more things, like for example, human hair, now it starts getting exciting because see, human hair, again, does not undergo as much of strains, but you'll see that after a certain point, you get more or less a flat response. Spider's web, which most of you will think as one of the most elastic materials, starts actually competing with the stress strain graph for steel up here. Okay, but as soon as we add our favorite tissue, hopefully by the end of this talk, you'll be convinced that arteries are really fun. when you start adding arteries, and here's a picture of, in the bottom, what you see is that aorta structure that I told you about, which stretches to about 60 to 80 times. So here, we have to actually test it in two different directions, because as you blow a balloon, it has stresses in the hoop direction and in the other axial direction. So if you plot the graph in both these directions, you get different curves. So this material is what is called anisotropic. So it will give you two different kinds of properties depending on which direction you're looking at. So it's a lot more complicated than a simple material like steel. Okay, and I've taken the same data and I've put it on a log-log plot, so you can see the initial part of the curve much better, of all these data much better. And again, you'll notice that the artery, regardless of what you do, is highly nonlinear. And so the theory and the analysis for how we handle tissues, biological tissues, is a lot more complex than that of materials like steel. So if we now compare what is the source of elasticity and how is it similar or different between metals and these polymers or biological tissues. So here's a cartoon of what we will non-dimensionalize as a unit for a metal. So here, each of those balls will represent one atom, and it sits at the corner of, in this case, it's an FCC lattice structure, which doesn't matter. But essentially, the idea is when we have two overlapping clouds of these atoms coming together, then if you push them too close, then you get into the repulsive domain. And if you take them too far apart, then you have attraction. So there is a sweet spot, which is the equilibrium distance at which these atoms are sitting. So every time we are pulling on a steel or any other material, what we are doing is essentially we are prying these two open. And so we're trying to pull those atomic clouds together, apart. And in this well, right at the bottom of the well is where that linear domain is actually present. So a lot of these ideas are based on using using these unit squares, and we try and understand what is the source of elasticity. But in polymers, we can't really come up with such simplistic ideas. Of course, there are ways in which one can do it, but essentially you have a thread like this, like a spaghetti. And if I were to hold the ball, blue colored section at one end, and then I start pulling on it, I have something which is completely entangled and coiled. And as I'm pulling it, I'm releasing all these coils and I'm stretching it out. And that is the restoring force which is put in these kind of proteins. So the whole way in which one thinks about what component of the energy contributes to this modulus, in metals it's dependent on the enthalpy terms which are present, whereas here we have something like an entropy which comes into being. And so essentially we are looking at how the different conformations of this polymer chain can actually work and contribute to the energy. So going back, why do we call it rubber elasticity? So yes, rubber elasticity started because in about 30 to 40 is when a lot of the people in South America and Southeast Asia, they discovered that this latex that you got from, this is one example of the tree, the rubber tree, and we call it gutta percha. It comes, I think, from the name of this rubber tree. And what they do is they extract this latex, and sometimes they would dry it out and saw that it was very stretchy, except that it wouldn't work for us, because if you increase the temperature, this was not a material that you can use in your labs or in your normal life. So then what they did was they realized that the reason why you could not use this white liquid is because it would just flow. So it was completely not cross-linked. So they introduced sulfur, and these balls are essentially holding the chains together at specific locations through a process called vulcanization. And what vulcanization did was it allowed this whole liquid to transform into a material that we can use as a solid. And so they could use it to erase marks on paper, so they called it rubber. And you could use it for many other things, like these very nice rubber tires, which we use frequently. And this becomes a material we can use only because of the presence of these really important crosslinks which are present. Otherwise, the liquid that you get is actually not so usable in our day-to-day life. So the discovery of this rubber proteins also ties back, and I forgot to mention that, I think I have to go back. Let's see. So with this discovery of rubber elasticity, a lot of the rubbers that we got in the 40s were because of the rubber tree. But because of the war which was going on, they realized that they could not have enough of this liquid which was coming from Southeast Asia. And so people started trying to make rubber-like materials in their labs. And typically there was one big company called DuPont which started out trying to make synthetic rubbers like butadiene rubbers, nitrile rubbers, all the things that we use presently. It all started because there was lack of raw material because of the war that was going on. So maybe war has some positives that it can create it can induce some creativity in us. But around the same time, there was this person called Thorkel Weisberg. Weisberg was a big hero of mine as I was a graduate student because I read all the cool things he did. So he was a scientist in Denmark and he moved to Cambridge and he was actually studying how locusts fly. And he realized that while he was studying how locusts fly and he was looking at the aerodynamics of it, he realized that for these insects to be flying all these, these are the challenges that locusts fly about 8 hours a day for 30 days, and that's 20 million cycles that they have to do, sometimes without eating. There are also other insects like cicadas who are singing at 4 kilohertz, sometimes 2 to 3 hours, so you hear it in a forest, that all the noise that they make. So they will sing continuously for 20 days, and that's again 400 million cycles of loading or unloading that these materials have to undergo. So he realized that structures by themselves could not be doing allowing the animal to do this without the presence of some energy storage component. And so he started trying to discover where is this located. And some of the most classic work came from his lab in 1961 to about 70s, when he started looking across different kinds of insects. And he noticed at the base of that, and this is not his work, it is a more recent paper in 2024, but it's the same locust. And so I picked this as an example. So at that wing hinge, at the base of that leg, you'll see this blue colored protein. And so he discovered it's a very small pad which sits there, and he was able to characterize it. He saw that this was a colorless cuticular protein. So the cuticle is made up of chitin, which is a sugar-like molecule, and it can go through various different properties, all the way from very hard, it's almost a 2 gigapascal property, all the way down to rubbers, which is about 1 megapascal. So that's the range of what the cuticular properties go by. But he discovered that this very colorless protein, it can actually be stained with methylene blue, and it looks blue. It's if you shine UV light, it autofluoresces. So that is one test that he said, okay, for us to find it in the cuticle, this is one idea. What's also interesting is it's very stable over a wide range of temperature. And it's hard and brittle when it's dry, but as soon as you put it in water, then it swells and it's able to do its large deformation. But it's extremely delicate. So it can be dissolved very easily if you put it in any of the enzymes, and it has a very delicate structure. And so this is a nice picture of the structure of the tendon, which is at the base of the dragonfly wing. So if again you see dragonflies which are hovering, it's able to do this really nice task because of the presence of this blue colored protein which is sitting over there. So he discovered it and he characterized it, and this is the first protein that one knows how well one person actually started with an observation and went all the way up to figuring out where it was present in many insect systems, showing us how to characterize it, measuring its mechanical properties, also trying to understand whether it has a structure in terms of a secondary or any other kind of structure. But he was not able to crystallize it. In fact, no one has been able to crystallize it, which is telling us that we don't have a structure for these amorphous proteins. And that's the idea he's proposed, that it's actually an amorphous network similar to the spaghetti that I showed you earlier in some of the slides earlier. What he also saw was that when he took these tendons of dragonfly and he stretched it, there were no strain rate effects. So even after several weeks having it stretched out, if you take a rubber band and pull it and keep it, after some time, the length of rubber band actually increases, right? So unlike that, this thing went exactly back to its original dimensions, which tells you that it's highly elastic. And he was able to measure the resilience, or the amount of energy that is given back after you remove that deformation. And he showed that it's almost between 97 to 99%. So just to tell you, broadly speaking, what resilient means, if I make a ball of that protein and I hold it fixed and I drop it back, then essentially my hand doesn't have to travel too far down to catch that ball. It pretty much stays where I am because that's the energy which is coming back. It's a simplistic idea, but it more or less tells you what resilience works like. So this is much better than even the rubber that we got from the tree, because that has a resilience of about 85 to 87%. So here you have a system which is not really undergoing any friction, and all that energy that you give by compressing it is given right back when you release that compression. So he's called resilin an extreme type of structural protein. So something is linked between the entropy of that whole set of chains which are present inside that protein and rubber elasticity. So he set up to try and study that. And so meanwhile, in about 50s, a lot of the theory work in continuum mechanics by people like Rivlin and Treloar and others had come into being, and they had built a really nice theoretical framework on how we should study rubber-like materials. What we knew was, if we imagine that I hold one end of the chain fixed at origin, my job essentially is to find out when I pull where the other end of that chain is, because it could be all coiled and so on and so forth. So if I'm able to find that distance r, and I imagine that it's made of n number of blocks, each of them of the same length a, then the effective distance r scales as n a square. So it actually is not, so what it tells you is when I pull it out, is when I would imagine that r is maximum, but actually it goes as square root of n times a. So what people thought was it's similar to a Gaussian distribution of different kinds of chains. And if I'm able to do this for a single chain, I can imagine a whole block made of these kind of chains. And now I can imagine the block being either stretched or pushed. And my job now essentially becomes trying to understand the distance between 2 points on this block and translating it to what I know about the single chain. So with this idea of entropy dominating the uncoiling of proteins, we can come up with relationships for how the stress, sigma, is related to the stretch being the change in length the changed length divided by the original length. And you'll see again, it's a very non-linear relationship. It does not scale as what you would have studied in high school as sigma equals E epsilon. So now you have a much more non-linear relationship, which is how we are able to capture how rubber-like materials work. What Weisberg also did was he showed that the elastic force, he looked at what happens to this rubbery protein resilin that he extracted from dragonflies, and he showed that you can change the temperature of this thing, and he plotted the force with temperature, and he was able to, the stretch lambda is new length upon original length. So it means I have stretched it to almost 40% or 39%, all the way to doubling the length and more. Regardless of that, he showed that the force can be measured and it scales like what he's plotted here. So if we ask the question, what is the force, how is the force related to this stretch? The red curve shows how we have measured it in an experiment. And through our models, we can calculate how much amount of that force is coming due to the entropy changes, which is ds by dl, or by the internal energy change, by doing these experiments very carefully in a small calorimeter. And he showed that essentially the changes in internal energy is more or less flat. And these are experimental data, so you see a little dip in that curve. Whereas the blue curve seems to hug very closely the red curve, which tells you that the entire mechanism of elasticity in such proteins is purely due to entropy. So this was one of the first systems that was this well characterized, and it fascinated people. And they went on to find a lot of resilin, not only in these structures where they fly, but they also discovered that there could be small amounts of resilin which is present in eggshells. It can also be present in the timbals of cicadas as they sing, and various other places. With that, I want to move to the next idea of what do we know presently about the rubber proteins in mollusks. And I showed you this picture earlier, okay? But just to give you a perspective of why this is important. So this is an animal called Kolbeki, and it lives in Antarctica. And in Antarctica, the water temperatures are extremely cold. It can be as low as 1.8 degrees Celsius, and which means that water is a lot more viscous at these low temperatures. So the animal has to be very clever on how it actually moves through these very cold temperatures. What's also very impressive is that a lot of materials that we use at such cold temperatures will not really work so well for cold temperatures. And just to give you an example, for some of us, we'll remember this as a very sad thing which happened, which was the Challenger in, I think, 1986. It burst into flames and it killed all the people on board. But people tried to understand what was really wrong with the Challenger and why did it burst. And they discovered that has anyone seen this or heard of the Challenger? Not the younger ones, but maybe the older people in the audience must have seen this at some point. So there's a very famous video of Feynman, who was tasked with trying to understand what went wrong with the Challenger. And in this video that that is present online, you'll see that to demonstrate what went wrong, he found out that it was actually an O-ring, a really simple rubber O-ring, okay? And the rubber O-ring, what happened was it was cold that day when they had this the space shuttle takeoff. Two was something made the rubber O-ring come into contact with very, very cold temperatures, maybe, I don't know, liquid nitrogen or something. But suddenly the rubber, which was supposed to be nice and elastic, suddenly came into contact with something cold, and that material changed its property from something which undergoes large deformation to something extremely brittle. So a lot of us are sometimes sitting on chairs where somebody has kindly left a small piece of chewing gum, and you'll get your dress full of chewing gum and you don't know what to do with it. So it's nice and soft and compliant, but how do you get rid of it, right? So as much as you rub, it won't come off. Cold temperatures can do the job because, it changes the material property and puts it through something which is very, very brittle. So that's what happened to the Challenger. It actually failed because it was this very small rubber O-ring which came into contact with cold temperatures, and then it was all chaos. Now that I tell you about Challenger, think again about this very humble scallop which is swimming in very cold temperatures, and temperatures are extremely cold. So we have a case of how if you decrease this resilience of this protein, it means it will increase the damping coefficient and it will not create enough power that it needs to provide the thrust that it needs to move. It looks similar to a bellow that we have, that you can move it back and forth and get it to move. So a lot of it will not work at cold temperatures, and yet this interesting protein called abductin, without which the scallop is not being able to actually move, can work regardless of how cold that temperature is. So again, people have shown that this block of protein, again, it's a really small amount, it's amorphous, it's again cross-linked, it's again plasticized because it has some water. It has a resilience of about 90 to 91. It's not as good as, but it's still much better than anything we've been able to cook up. And it again acts as a compression spring. And so we can get into details on what is the kind of crosslinker, and almost all the crosslinkers that we have in both resilin and abductin are derived from tyrosine, for some of you who are interested in the biology aspect of it. And this is a proposed model. So there is at the hinge shown in that circle, you have a muscle which is trying to pull the two scallop mouths together. And at the base is this resilience element which is present. And as soon as you compress it and you let go, that allows it to push the water out and move wherever it needs to move. Unfortunately, not much is known about abductin. So even if you try and find, there are hardly 5 to 10 papers that you'll find on topics such as abductin. One is because of the scarcity of where to find these animals. Two is maybe there's not enough interest and it's a small protein, but there are hardly any papers on what is the mechanism of elasticity and others. We don't have much information about this. So it's an open place for a lot of us to actually think about contributing. But again, the third class of rubber-like protein is elastin. And here's a picture from our lab where you see all the black color squiggly lines. That's the protein rubber, and that is elastin, which is coming from an artery. This is not human, this is from a cow artery. But regardless, we see similar kind of elastin architecture in humans as well. So I'll talk a little more about this elastin, and yes, there's lots of data on this because of its link to disease. And going back again, that was the structure I talked to you about. And so we like to construct pressure-volume relationships to understand how efficient the heart is. But one of the problems is as we get older and older, the arteries start stiffening. So even if we don't have plaque buildup, we have what is called arteriosclerosis. And that happens because a lot of this elastin gets broken down. And the elastin gets broken down and it's not efficient as an energy transfer. So all that stretch that I told you, it will not work. So if you have a lot of resistance built downstream, then our Windkessel, which I told you earlier so the heart, from there it goes to this elastic artery, which bulges out. And so you have some amount of the Windkessel action created as a marriage between resistance and compliance units. So if the resistance increases downstream due to peripheral arterial tissue properties, then you actually load the heart much more and we get all kinds of disease. So it's important to understand what this protein elastin is doing in our vessels. Just to tell you, arteries are a lot more complex than most of the tissues. So there are this pink colored layer of cells which are called endothelial cells. They align in the direction of blood flow. The media is comprised of a lot of elastin, and there is almost about 50 to 75% of that elastin which is present in structures like the aorta, which is the largest vessel in our body. Adventitia is a tissue which is primarily made of collagen. Collagen is another protein, but not as rubbery. The collagen structure was proposed by G.N. Ramchandran, who was at IISc and started the MBU unit. So if we pull arteries, we get this red colored curve, which is again a nonlinear curve. But what's really interesting about this nonlinear curve is that the initial part of this stress strain curve is elastin dominated, which tells us that with even a small amount of load, it stretches out a lot. Whereas you don't want your tissues to keep bulging out, right? You can take your skin and pull on it, and you'll see it's initially easy to pull on the skin, but after that it becomes more and more difficult because of this protein called collagen, which kicks in at some point. What's interesting is this transition between elastin to collagen-dominated response is where the physiological operating pressure of an organism normally sits. So the presence of elastin is extremely critical because if we get rid of it, you shift the curve towards the y-axis and you make it stiffer, which again tells you that it puts a lot of load on the heart. So it doesn't work out. What's again interesting is that each of these units, so the blue colored things over there, they're not eyes, but there are smooth muscle cells. They are very closely associated with the elastin protein, and they have a bundle called smooth muscle cell, elastin, and collagen, and that is called a medial lamellar unit. And the number of medial lamellar units seems to be conserved across various species, and each of them bears a constant amount of load. So it tells us about how evolution could have happened, and I'll again tell a little more about this later. But again, is elastin very similar to that of rubber proteins? The answer is yes. There are again cross-linkers, and these cross-linkers are slightly different from what was seen earlier. These are called desmosines and isodesmosines. They're again highly insoluble, which means even if I were to cook this at very high temperatures, and the way we isolate this protein is we cook it in water or sodium hydroxide at almost 100 degrees Celsius. And everything else comes out in solution, but you get a nice yellow colored sheet of this rubber protein, which is very, very fragile, but very stretchy. In humans, the gene for making elastin, it's a set of genes, it gets turned off at puberty. So the amount of elastin we have is pretty much all we will retain for the rest of our lives, except in one case, which is in women who make a lot of elastin in their uterus during pregnancy. And again, it gets degraded as soon as the baby is born. And in cases like cancers, where sometimes you see elastin being deposited. So what is really exciting is that, that the amount of elastin we have in our arteries should last us for whatever number of years we hope to live. Let's hope 100, right? So that is the challenge that this protein faces, to constantly undergo this stretch and do this 72 times a minute for 100 years. So how is it able to achieve this fantastic task? So people tried to show that it was important by knocking down elastin, which means that your cells and the body does not have the ability to make this protein. And the baby wasn't even born, and these are baby rats, but they died in utero, which tells you that it has a very critical role in during arterial development. So without elastin, we won't be here, is what I want to sort of say. So if we compare this, again, this is a picture to show you the nice, to show you the elastin structure, which is all coiled. And so you'll see that the green colored structure is all present as coils, and it's connected to the smooth muscle cells in red. So again, 55 to 90% of thoracic aorta is all elastin. And it tells us how important it is to act as that Windkessel element. Again, it undergoes really large deformations, typically about 80% stretch during each cycle. It stores energy in the systole cycle, and it returns it during the diastole. Pure elastin is again very brittle and rigid, but it requires water as a plasticizer to allow us to see its large deformation behaviors. Aged and diseased arteries have much reduced water content, and they also have many fragmented elastin. So it's not able to do the load transfer, which is its primary job inside the vessel. So people are trying to look at how this elastin is made. So it's again made and it's transported from the endoplasmic reticulum through these vesicles all the way outside. And it's thrown outside the cell. But there you don't have just the pure elastic protein. It seems to sit very closely with another microfibrillary network, which helps align the elastin properly. And there it is cross-linked. Once it's cross-linked, it becomes insoluble. This part is extremely important because the fibrillin seems to contribute a lot. And there are diseases associated with fibrillin, which I'll tell in a little bit. And so the fibrillin with elastin makes it a very potent combination. People have tried to study what the fibrillin structure looks like, and these are atomic microscopy images of fibrillin. And what they see is some structure which looks like beads on a string. And as you pull these beads, which is shown in this picture here, these structures uncoil, and that gives it its elasticity. This recent picture is one of the most exciting EM pictures taken very recently, about 2 or 3 years ago, which is the first time we've seen what that microfibrillar network actually, single strand of fibrillin looks like in real life. So when we test tissues, it gets a lot more complicated than testing steels. Because if we want to pull sheets which are extremely thin, one is how do we clamp it? How do we view it? So we have to go back and still do the same order of kinematics to show that you have deformations which are homogeneous. We assume some aspects of things which I won't get into. But the final relationship is how is this stress related to strain? In one case, I told you it was a Young's modulus, but here it's a lot more complicated because the material is nonlinear. So here is one example of how we do our experiments, and this is through a instrument we have built in our lab. And so what you see here is a stretcher, biaxial stretcher, which is sitting on a microscope stage. And please pardon the movement. I think my student is moving his arms too much, but what you see is two sets of arms where it is clamped, and then we pull it and use the microscope to view how this stretch is happening. And this we can do this with with sheets of elastin. We can also do it with cells and so on and so forth. So this is, sometimes we hook it up like this in trampoline fashion because we have to control the boundary of all the four edges. So things get complicated very, very quickly on how to hold it, how to clamp proteins and so on. But this is the kind of data we get. We can pull back and forth and we can get strains in two different directions that we can analyze. So when we analyze that, what you see, interestingly, are these, I told you all along that elastin is nonlinear, or the arteries are nonlinear. But here we have properties of elastin, which is looking more or less straight, very linear. But you'll see the amount of stretch in some cases has gone as high as 80%. So you can do both these kind of experiments, and our goal is to develop models to better understand how this elastin works. Again, these are going back into how rubbers work and looking at the structure of how elastin is laid out inside the vessels to come up with a composite understanding of this. So I want to change track a little bit and tell you some in the last few minutes on how the mechanism of elastin comes about, elasticity comes about in elastin. It comes from very hydrophobic residues which are present. So resilin has 34% hydrophilic amino acids, whereas elastin has only 5%. So which tells us that it is a very hydrophobic protein. Single elastic fibers again have no structure. So you can find small units and you can get some structural information from small parts of it, but overall it cannot be crystallized. Again, it behaves like an amorphous rubber-like protein. NMR studies show that the backbone is highly motile. So this is so-called a simplified picture of how it works, and this is from a very recent paper using MD simulations, which shows that there are helical domains shown in magenta, and those are cross-linked together, and it's present in it's in the presence of water, which is shown in blue, and hydrogen bonds. All of these contribute to how they uncoil and stretch and contribute to the elasticity. But most of our understanding is that the hydrophobic aspects of the components of this elastin actually are very critical in driving the recoil of this protein. So when we now take data from host of different vertebrates, and here you'll see in this paper that they've got data from squid, lobster, toad, snake, etc., even a whale, that they've been able to test the properties of these vessels, you'll see that the modulus, which if you remember was the slope of the stress strain curve. So the modulus, it actually increases with the pressure, and this has been normalized to the operating pressure of that animal. But very interesting to see that almost all the curves fall on this straight line, right? What is also interesting is that the modulus of the aorta at this in vivo pressure is approximately the same for all the species, be it a rat, a human being, a squid, or any other creature. So, but when we go back and look at how elastin has evolved in vessels, we see that these black color lines here are histology images to show you where that protein is located. And we see that there are well-developed elastic fibers which are present in all vertebrates. But you'll see that in some animals like B and D, especially B, you see it's very fragmented. And lower vertebrates have more open arrangement of these elastin protein. and it's more concentrated inside that lumen or the tube where the blood flows. It's completely absent in lampreys and hagfish. These are jawless fish which evolved much earlier. These are very primitive vertebrates which happened, and it's completely absent in invertebrates. If you remember, the protein rubber of invertebrates is actually resilin. It's not elastin. So if we go and ask the question, how did elastin actually evolve? And it seems to start very early from when vertebrates evolved. And it seems to coincide with how the circulation system changed from open to closed. So as we moved into closed circulatory systems and we saw an increase in the operating pressure of the organism, for example, 120/80 or is where we work. So as we move towards higher and higher pressures, there was a need to develop such systems so you could do all that action which was required. So if we ask the question, when did elastin actually evolve in the phylogenetic sense? Was there a major event which happened? We don't know. Can we find a primitive form of elastin that points to a common ancestry? Are there any other phylogenetic changes in the structure that relate to some mode of evolution? Was there one specific event that changed how globular proteins change versus a change in how these kind of proteins evolved? The answers are not completely clear. What we know is that if we quantify a hydrophobicity index, and we won't get into details, but these are based on the different kinds of amino acid residues which are present. So you'll notice from right to left, so we go from lower vertebrates all the way to birds through, and again the mammals are there. So you'll see that the hydrophobicity seems to be increasing as we get into higher order animals. So they suggest that the evolutionary mode is very different from point mutations, and they are genetically different elastins. We don't know if there's a common ancestor from where this elastin started. And so this is the only paper that has actually tried to combine all of this data into one, and that is as old as 1979. But we still don't know much about why are the elastins getting more and more hydrophobic. One thought is that if we increase the hydrophobicity of this protein, then the protein can increase its modulus without really increasing its cross-linking. Because if you increase the cross-links, it means it won't allow that protein to undergo large deformations and do all the jobs that rubber proteins do. Because if you add more and more cross-links to rubber, it starts becoming just like a solid block. It won't deform a lot. So you have this sweet spot that you have to hit by trying to work at higher pressures, but trying to work with less cross-linking. And so the only way it seems to have evolved is to increase its hydrophobicity. Again, some clinical relevance, and this is a paper from a collaboration in Namrata Gundiah. That is the aorta, and it was a straight tube. If you see in this person, it's a woman who, and you see there's a little tear which is running down over here. Okay, so this is the ascending aorta, but it's extremely bulged out. It's not only bulged out, it's thinned out. And this person required an implant, which is called an elephant trunk, which was put over here. But the main reason why this person had this thing which enlarged was because they had mutations in that fibrillin-1 protein. So the kind of elastin which was present was very poor. And so this person, it's a very major surgery. And apparently a lot of people in Namrata Gundiah would come with such things. We are interested in studying the properties and also looking at the flow physics of how blood flows through such curved pipes, especially when it's got this bulge there. There are other diseases like this aortic stenosis or Williams syndrome, which has deletions in elastin gene. And so a lot of them have partial loss of elastin. And all these patients require surgeries because of the problems that happen in the valves or in the tubes themselves, which are conducting flows. Again, this is Marfan syndrome. So Marfan syndrome, for those of you who might have heard, Abraham Lincoln is supposed to have a Marfan syndrome. Very long extremities. Iknaton supposedly had Marfan syndrome. So it's not clear what gets people to have this. It's clearly a mutation in this fibrillin gene, but these people require surgeries at least a decade before most other people. So a large number of people have these Marfan syndrome and also have these aneurysms, which are, again, abdominal aortic aneurysms. Einstein died of an aneurysm in his abdomen. And these are vessels which will suddenly become enlarged and tear, and then blood, which is flowing at 1.2 meters per second inside this tube, is released, and that causes very quick death. So these are the main issues why people need to study proteins like elastin. So just to add, elastic fibers are large, complex, but surprisingly very little is understood about how the mechanism of its elasticity. The main challenge is to try and identify the temporal hierarchy and the different kinds of interactions that are present not only between elastin and the cell, but a host of other proteins which come into being. And so that is one of the big questions in science right now. But more importantly, very few studies are focused on the biomechanical properties of these microfibrils or the fibrillins that I showed you. And their molecular basis needs to be better understood. Other questions are, how can you measure these forces during deformation in tissues which are elastin rich? What is the mechanobiology, or how do cells and the elastin interact together and give us the kind of form and function that we see in normal tissues? So these are all present questions that the community is trying to understand. From a technological perspective, the more exciting things that people have discovered is that they have seen how to, they have discovered the genes which can be used that encode for pro-resilin. They've been able to clone and express resilin in E. coli, and they've cast it, and you see this whole tube of resilin, which you can make large components of just by photochemical cross-linking. So this, of course, was 20 years old, but I thought that was one of the most exciting technological advances that I had seen. I had to leave you with one thought on, this is a video from Flubber, and the person who taught us polymer science and engineering actually said that she made this Flubber because I come from Berkeley, where the colors are blue and yellow, and so she said she combined them and made this green-colored protein, green-colored rubber. And just to tell you that what we expect of rubber-like proteins is similar to what you'll see Robin Williams doing when he drops this ball. It's not going to actually undergo any frictional losses to the point when you can see it shoot all the way into space, right? So that is what we expect from rubber proteins. I think I'm slightly out of time, so I won't tell you some of the other strategies that animals use in biomechanics, but I'll just thank you, and I've tried to put a picture of most of the PhD or students in my lab, so they'll be happy. So thank you very much.
Thank you, Professor Gundiah. Now we'll, yes, sure. Q&A starts.
That was a very exciting, and you have to come back to this kind of. Can we replicate the properties of these proteins, elastic properties, for one mechanized mobility of humans? One thing. Then design pumps that can be used for pumping fluids like water, gas, petroleum, like that. Can we go deep and replicate for realistic applications?
So I think for pipes and others, we have cheaper options to work with steels. They work much better. But if you want to talk about what is elastin actually doing and why do we need this? So a lot of the work is going into making materials that can be replacement for our arteries and for skin and other replacement. Skin has very little amount of elastin, about 3% or so. It is still extremely critical because it gives it its elasticity. So if your question is, can we make synthetic elastin? We have not been as successful in making such things, but they're trying to use it to make wound dressings or make tubes of elastin for growing cells and making a replacement, because when they do these bypass surgeries, they always take tubes. But the idea will be to take normal tubes of elastin with our cells and put it as replacement in. So that is still the holy grail. It has not been done, but people are trying to make such tubes. Hopefully I answered your question.
Thank you. It was an insightful talk. Is the evolution of elastin, is it a adaptation, X adaptation? Was it?
Your question is it an adaptation or?
X adaptation? The elastin is a X adaptation. Is it you have taken it from previous one papers?
So your question is, when did elastin evolve? So there is, like I said, a single paper by Sage and Helen Sage, who has talked about evolution of elastin. I think there's a lot more that needs to be done to identify this common ancestor, like what I'm telling you, is at which point during the evolution tree did we see elastin come about. So the only connection we have is that it seems to be present in all vertebrates, even early jawless fish. The only quality of elastin is very different. So like I said, although we call it elastin, there is a change because it's becoming more hydrophobic. It is still elastin, and it seems to coincide with closed circulatory systems. That is the only connection as far as the evolution process is concerned. But I think there is a lot more room to study some of these processes. I think it has not been on the radar for many people, and that is the only paper that I have seen that you can go and find if you're interested.
Can you please show that video of water pulsating through the pipe? And
that was
how
a nice video, no?
Yes,
I thought it was fun.
Yeah, thank you.
Sorry, let me just go to my. Yeah, this one, yeah? Right. So you see that it's a nice high school project to demonstrate this Windkessel action, which a lot of us are not aware of. But I think the whole aspect of energy recoil in a little balloon is very nicely captured in this video.
That one property, can it be replicated for transportation for humans? That flea jumping 150 times, can it be replicated, redesigned for
humans?
For mobility, I mean transportation, some sort of.
You mean how can we use such proteins to translate for human
on a large
mobility?
scale, we design it doesn't lose energy.
Right. Well, if we engineer it, we are left with some of the best known rubbers. Like I said, some of the nitrile rubbers and others, or butadiene, styrene, butadiene rubbers are pretty close. But will they give us 150 times? I don't think that is achievable. One thing you have to remember is people like us, we also want directional ability to be able to control our movement, not like a flea is jump and land, hopefully in a good spot. So if we think whether technologically we'll be able to use such jumping ideas, I don't know. It's not so clear. Yeah. So that is the goal in understanding jump ability of insects, right? Because it's not extremely well studied. In fleas, it has been studied. But there are a lot of insects which take off and jump from much different substrates, like what some of my colleagues are trying to study in biomechanics is how do insects jump off leaves? Because, when you push down on a leaf, you don't get that ability to push. So how is that jump actually translating in a in an insect on a leaf? So those are some of the questions. If we understand better, I think we might make, as far as robots are concerned, I'm not convinced I'd want to be the guy jumping using this contraption. Right.
Thank you very much, Professor, for your brilliant work and wonderful presentation. My question is regarding this one equation, one vector R, which you showed as n equals a square.
R square equals.
Actually, in this case, you have shown the two endpoints of the string.
Yes, yes.
But in a mesh, when everything is meshed up, the endpoint doesn't make much difference.
You're right. So this is sort of a very nice question. It's a thought experiment that one does, is to think of holding one end fixed and thinking of the other end where it is located. So the point I was trying to make with this is we are never able to see the endpoints of both ends of the polymer. You're absolutely right. But what it gives us is a theoretical analysis. For example, if I were to take this gutta percha or polyisoprene, which has units of isoprene which are same but stacked one over the other, which is like made a polymer out of it. So if I'm going to use that analysis, I'll think of this room of people and what is the height that we look at, right? So that is why we are thinking of a distribution of what these ends look like. You're right, I won't be able to see it, but what we do...
Probably, is it an isolated string? You have separated?
Yes, so what we do is we think...
only one string.
Yeah, we have not taken it experimentally. We have theoretically analyzed it for one chain only. What we do is we assume a rule called affine transformations. We assume that if it's called affine, A-F-F-I-N-E, affine transformation. So we assume if that is present on a single chain, I should be able to apply. So if I were to apply it on a cube, like what I showed, the same idea can be translated to a single chain, right? So whatever dimensional changes I see in a cube, okay, I could relate it to what happens in a single chain. And there the analysis works very well.
So this affine transformation, is it what sort of, is it something like a wavelet or?
No, it's not like a wavelet transformation. It makes rules about how the deformations work at a single 1D level to 3D level. And it says if I see the distance between in a blob is this much, I can relate it to what happens between the two ends of a chain.
I got it.
It just helps us think about it. But you can't really see any of these features that I'm telling you.
But by getting entropy, you're making it more complex. No, what do you actually mean by entropy here?
So entropy in this case is the change in conformation of that chain. So if that chain has, so we know S is equal to K ln omega, right?
Correct.
So if I were to look at the probability distribution of these chains and tell what omega looks like, I can tell what the entropy is. Okay, and relate the entropy to force through that relationship because it's a thermodynamic relationship. So here it gets even more complex because now the thermodynamics and force is being related, whereas all along we didn't relate those two except in rubber-like materials. We do that. And that is the theory which is, it's not mine, it is from 1970s that it has come about.
Probably I'll come back to you offline.
Hopefully I answered, otherwise I'm happy to talk to you after this.
Yeah, thank you very much for a very insightful talk, as always. So I had just two questions. So all the examples that we discussed about flexible proteins, they have been from the animal world. So do we find similar flexible proteins in the plant world as well, and what are their functions?
That's a really nice question. So the only example I see in the plant world is latex, which has been used extremely well for the gutta percha, rubber-like materials. It does pretty well if only when you cross-link it. But here in the animal world, it comes pre-cross-linked, right? So that is why it is structurally useful. I don't know why plants make latex and what is the end goal which helps them make the latex. We have been able to leverage it for our engineering applications by vulcanizing that material. So I'm not aware of many other examples, but I know there's many other plants which make this sap-like latex substance, but none of it is usable unless you do a chemical process to treat them.
So there are no functional attributions to these.
So we don't have the classic rubber. So like I said, if I take the latex and I vulcanize it, then I can say, yes, it's a functional use for me. But what is the use for a plant is what we need to ask why it's making the sap. And that is not a rubber protein in that sense.
So other than this latex, we don't know of any other flexible protein?
I'm not a plant person, so I don't know properly what the other examples are in nature. Right. It has, no, I don't think that does not work. So those are touch sensitive, but these are energy storage structures. So the mechanism is very different. Huh. Right. So there are no other examples. I want to clarify, no other examples of rubber-like proteins in the plant world, to the best of my knowledge.
I had a second question. So I had read something about the scallop theorem. So according to which a scallop should not be able to move at all. It takes in water and then it pumps out water, and so it should stay put in one place. But we see that the scallop actually moves.
It actually moves, yes.
So is it because of the deformation in that protein that actually binds the two halves of the
Right. So there's two parts to the question. One is, how does it move? It will not move without the presence of a muscle, which is absolutely essential, which connects the two valves together. What is important is the quickness or the speed with which it moves. That is related to the hinge rubber protein, which gives back, because as soon as you compress it and let go, all that energy is given back to the scallop. And of course, there is water movement, because as you push and release, the water goes there and then the animal goes there. So both parts have to happen, otherwise it will not work. So yes, so the animal, well, I have not thought as much about how how uniformly or the valves to open. So it's a little more complicated because of the jaws opening and the water being pushed as well. So there is that interaction as well which is happening. So you're right. I think it has, and consistently when we see the videos, the animal doesn't move straight. It sort of does a little bit of that, but it's a complex environment as well. So I don't know the answer to your question.
Any more questions? Okay, in that case, let's wrap up November's Kaapi with Curiosity.
Thank you all.
Thank Professor